All right. Welcome, everyone, to the CHIP Intelligent Health Lab. Today, we're going to hear about real-world evidence for a learning healthcare system, moving from claims data to claims with linked EHR data, from one of the world's experts in real-world evidence, Sebastian Schneeweiss, who is a distinguished professor of medicine and epidemiology at Harvard Medical School. I'm Ken Mandel. I direct the Computational Health Informatics program at Boston Children's Hospital. We're a 28-year-old multidisciplinary applied research and education program, and we host the Intelligent Health Lab series, which features thought leaders across healthcare, informatics, IT, innovation, and more. If you want to follow along on Twitter or tweet, we're at boss_chip. I'm going to briefly introduce Dr. Schneeweiss. He will give his presentation. You can put your questions into the Zoom Q&A box as we go, and then we'll have some closing remarks. So Sebastian, who is a close friend and colleague for many years, is a professor of medicine and epidemiology at Harvard Medical School, chief of the Division of Pharmacoepidemiology in the Department of Medicine at Brigham and Women's Hospital. His research focuses on assessing the effectiveness and safety of biopharmaceuticals in clinical practice. He is extremely well published, extremely well funded, and he runs very important projects that you'll hear about today, including the FDA Sentinel Innovation Center and the RCT-DUPLICATE initiative, which is designed to understand when and how real-world evidence studies can reach causal conclusions and how those get fed into a regulatory process. So to get brought up to the very bleeding edge of the use of real-world evidence in the assessment of therapeutics, let's turn it over now to Sebastian. I'll stop sharing and off you go.
Well, thank you very much, Ken. Really appreciate the invitation to speak here. And I have zoomed in the title a little bit more, and what I'm doing for the last 25 years is all around producing generalizable evidence on how safe and effective medications are in clinical practice. Now, over the last 5 years, 5 to 7 years, the FDA got massively involved because of the 21st Century Cures Act that forced them to focus on thinking how real-world evidence, how the use of healthcare data that are getting generated through the routine operation of the healthcare system, these are claims data, these are EHR data, these are medical devices data, how will they fit in their decision-making process in approving medications, in the surveillance of medications with regard to their safety. So this production of decision-ready real-world evidence is key and center to all of that. And I will talk a little bit about reproducibility and validity, two core concepts that lead us along that journey. By my conflicts of interest in my disclosures, I specifically disclosed that I am co-founder of a company called Etienne, and I receive consulting fees because that software was involved in the conduct of the study. Now, a few years ago, a whole bunch of regulators and health technology assessment representatives came together and tried to decide what's the characteristic of decision-ready real-world evidence, and we came up with these 4 characteristics that are really easy to agree on because they're fairly high level. You want to have meaningful evidence where the data fits the problem and the right problem is answered. You want to have valid evidence. You want to come to causal conclusions whether a drug is causing a certain health outcome. You want to have the evidence expeditiously, because if you don't make a decision, the status quo is the decision. You do make a decision, but you just make an uninformed decision. And you want to be transparent. Transparency comes before validity, because if you don't know what was done, you cannot make an assessment about the validity. And in this whole game that we call decision-ready real-world evidence, you always have an external stakeholder. We, as academics, we produce evidence, and the consumer of that evidence is the United States FDA, the European Medicines Agency, the guideline writers of this World Health Technology Assessment Company. So we have to be transparent in what we are doing so that other people, other stakeholders of our research, can make an informed decision whether they want to trust our evidence or not. Let's briefly spend some time here. I call it a fairly fast clip here because we have a fairly educated audience. We all know one-off programming when we work with electronic health records, with claims data analysis, SAS data, R, what have you. When we do studies in our shop at the Brigham, where we know these studies will go to a regulator, we do double programming. Double programming means we have one protocol, we have two programmers, they're very experienced programmers. Every time we do that, every time we get two different findings. That's really unsettling. So something gets lost in the translation from the protocol to the programming code. And these are not syntax errors or things like that. These are misinterpretations of the protocol. And there's a litany of similar evidence that this one-of-line programming is a source of mistakes, of the wrong interpretation of the study protocol. And even if you share programming code, which looks like this here, and a person like me, or I would argue most stakeholders, cannot check whether the spirit of the protocol was implemented in the right way in the programming code. If you have the programming code and the data and you put them together, of course you get the same finding. And that's a very trivial hurdle of reproducibility. The much more important hurdle is whether the protocol was implemented the right way. We came up, the two professional societies, ISPI and ISPOR, who worry about these things, came up with the study parameters that need to be reported in order to make a real-world evidence study, a study on the association between a drug and health outcome, reproducible. And this comes particularly from the Sentinel Initiative, from the early years, and this is around now for more than 10 years, where we started programming macros where one macro is an entire study. So you feed in all the study parameters, and then you click run, and it runs, and you get the whole study done with one macro. Now, that requires that you specify, that you parameterize the entire study. And a whole bunch of parameters that you specified. And these are the parameters that we need to communicate to our audience, because these are the key study parameters. Without that, you don't have a full specification of a study, right? So this was the beginning, the Sentinel Initiative, and newer softwares around. This is the Edion software, where you have this bit more comfortable now. You select the patients, select the comparison groups, you select the treatment strategy, and as treated, and ITT analysis, and things like that. Follow-up model, the risk adjustment methodology that you use, some feasibility and diagnostics, and then a report. Now, this is following the target trial paradigm, like as if you would set up a randomized trial without baseline randomization, obviously, and without primary data collection. Now, what it also means, as you are specifying your protocol, you can go deeper. You can generate a statistical analysis plan from the parameter that you have specified. You can go even deeper. You can look into the plain English description of the variables that you have defined, and you can go one level deeper where you see the actual codes that you used. And this is what I mean by 100% transparency, from the study parameters down to the code level. You specify, you register your study on any of those sites, and you allow audit trails where you see exactly who manipulated data at what point in time. Was it before people have looked at the results or after people have looked at the results? It's a big reason of concern for regulatory agencies when people work with secondary data, that people might sniff around and just run 10,000 analyses and pick the one that they like best. So these are all ways to combat that fear. Now, when we put this all to work, this was a project by the Arnold Foundation that Shirley Wang was leading, identifying 150 published studies, published database studies, using 6 different data sources from the United States, from the UK, claims data, some EHR data, We were using the same data sources in order to reproduce, not replicate, but reproduce the same finding given the instruction set that was communicated in the publication and on a public web page. And what we find is that everything should be on the green line. These are the 150 studies on the x-axis here. Everything should be on the green line, but it's not. There's some outliers where we really need to understand why, what was not communicated to the stakeholder. In this case, we were the stakeholder, so that we got these very different findings. And that's reflective also in the point estimate, in the treatment effect estimate, where everything should be on a diagonal here. And you see a decent correlation coefficient, 0.85, between the reproduction and the publication. But still, look at these red dots. We highlighted just the 10 strongest outliers. What was not reported that we came to such different findings, although we used the same instructions as they were published, right? So there's plenty of room for improvement, and this all fed into this START RWE initiative, kind of solidifying the parameters that we all need to report here. The regulators were now very much involved, the 3 key regulatory agencies, which in turn led to a harmonized study protocol that is, I think, getting binding now. So this is the European Medicine Protocol. This is the Professional Society. This is FDA. And START RWE. And at the end here, you see this harmonized study template that's covering all the different aspects that you see in the very first column here, in a mix of structured information in tables and free text information. And this seems to become binding in the European Medicines Agency. We have to see how this fares in with the other agencies. I just wanted to give you a glimpse in the seriousness with which we are driving this enterprise in almost catching up with the world of randomized controlled trials, where this rigorous transparency has a much longer history. And if we want to be taken serious by decision makers, by serious decision makers, we need to be serious about transparency and reproducibility, so that everybody can understand what we produced and what we demonstrated as evidence. Now, building on that, we now can worry about the validity of these non-randomized studies using secondary data. This is where RCT-DUPLICATE, in a more narrow sense, comes in. It's a family of studies that aim to understand and improve the validity of real-world evidence studies for regulatory decision-making. It was funded by the FDA, post-hoc also by NIH and Boris Wellcome Fund. The aim number one, and the key aim really, was to emulate 30 RCTs and predict 7 other RCTs that were ongoing phase 4 trials. The key learning was specified as, had we replaced an RCT with a single similarly designed real-world evidence study, would FDA or would a regulator have come to the same decision? So that is a very narrow question that we are trying to answer. There are many other questions around this, obviously, but just imagine instead of running 30 randomized trials, pharmaceutical companies, these are all pharma-sponsored trials for approval, they would have submitted real-world evidence study, and the regulators need to know, would they have come to the same conclusion? So this is at the core of RCT-DUPLICATE. And this is getting expanded. We've developed a process, and then, of course, more scientific investigations based on that. There's a huge team involved. That's the Brigham Harvard team, is the faculty, a large staff involved in, thinking about it, in 37 real-world evidence studies over a period of 3.5 to 4 years. It's quite a task. The Edion team, our colleagues from FDA, with whom we conferred, every 2 weeks in order to go through the many questions that came up, and an advisory panel of great insights. Much is published already on this, and you will find it in the literature. So just as a reminder, what we're trying to do is, there's an actual trial that was completed, 30 trials that were completed, and we want to emulate them in a database study. And for all pragmatic reasons, this part of RCT-DUPLICATE is entirely with claims data. In subsequent parts, obviously, electronic health records, I'm going to talk about that as well. Now, the problem is if once we emulate the same inclusion-exclusion criteria, the same analytic strategy in a database study, we might emulate a trial that's actually slightly different from the trial that we wanted to emulate. Ideally, these two block boxes would be overlapping, and then we would be emulating exactly the trial that we had identified that was submitted to FDA, right? But that is not guaranteed. The infrastructure that we set up is a fairly linear one. We identified the data sources, whether data sources are adequate for the trials, is the data quality fit for purpose, and if not, we exclude those trials. Develop a statistical analysis plan, do some feasibility analyses. If problems seem to arise here, we don't go forward, because if you would be a company and your asset is at stake, you also wouldn't go forward because you're underpowered or you have residual imbalances and things like that. Only then do we register a protocol, run the analysis, structured report goes to FDA. This is all done within the platform. And the advantage of that, of course, is that we allow the FDA to use the same platform. The data remain on AWS, on the cloud, but we share the data analytics platform with the FDA so that they, given the regulatory considerations, they can make changes to an analytic plan. They can change the outcome definition. They can change the risk adjustment strategy because they disagree with our choices, right? And this circle is enormously powerful because it gets around this paranoia of worrying that people have picked the best analysis. Because if you pick the one best analysis out of 10,000, this will become obvious very quickly once you change some of the parameters and things fall apart really quickly, unless you have very robust findings. So it's very powerful to be able to share the data analytics platform. Now let's jump right ahead. Here are the findings. These are 32 RCT emulations, 30 of completed trials and 2 of predicted trials. Here you see the log of the real-world evidence hazard ratio, and here you see the finding of the RCT, right? And ideally, these dots would all be spec on the diagonal, right? What you see, this is record 1, has this enormous effect size way down there, and we do quite well in emulating this. But it's kind of out there. The cluster is around a mild protective effect or no effect. The blue and the red marking indicate that some of these trials are easier or better to emulate, and other trials are much harder to emulate. Blue is well emulated trials in their design, in their measurement, while red is we had some issues. Overall, the correlation is 0.8. Isn't bad, but isn't good either, right? We would expect more. However, if we can emulate the trials well in design and in measurement, then the point estimates are also very close. We now have a correlation of 0.94. So we're much closer here, and it is quite satisfying. Now, the issue, of course, is these emulation differences. There can be differences in population. Although we apply the same inclusion-exclusion, we still get different populations. And I'm going through examples for all of these. The treatment strategy might not be emulatable with the granularity of the data that we have. Placebo is something that we don't have in clinical practice. Outcome ascertainment and follow-up assessment, there are difference in the measurement, the difference in the surveillance strategy in clinical practice versus an RCT and things like that. So these are all issues that can arise and make the randomized trial different in design and measurement from the real-world evidence study. So that is a nuisance. We want to get rid of that. What we really want to study is the bias, right? That's what we're really interested in. Is the residual confounding because of the lack of baseline randomization? Our outcome ascertainment, was that complete, or was our surveillance metrics, was that insufficient, right? And the same for follow-up. So you see the tension, right? Only if we can emulate a trial well, can we make a conclusion, this very narrow conclusion, whether a real-world evidence emulation is valid or not. This is actually splitting up the blue from the red, the blue one with good emulation, the red one with not so good emulation, and here you see 0.93 versus 0.46, the correlation coefficients. Now let's dive a little bit deeper now. Here we have, I'm going through these 32 trials now. This is the first pack of 11, where we have the diabetes trials here, and some of them might be quite familiar. On the very right side, you see yes and no, whether the emulation was close or did we have difficulty. So the green yeses here means that we were able to emulate the trials quite well. You see here the RCT point estimate, and you see the real-world evidence estimate. The standardized difference is a standard error adjusted metric of the difference between the point estimates. So this is scientifically speaking actually the most interesting metric, I would argue. We have a bunch of binary agreement statistics, regulatory agreement, estimate agreement, and things like that, and that might be interesting to some stakeholders. Then we have 3 interplatelet trials: Triton, TIMI, Plato, and ESA React 5. The next batch is the direct anticoagulant or oral anticoagulants for atrial fibrillation, Aristotle, Rely Rocket, and DOAC for VTE, the Einstein program, Recover 2, Amplify Record 1, and hypertension treatment, Transcendent on Target. And again, always here the RCT finding, and here the fully adjusted real-world evidence finding. And here the standardized differences. And here, for example, you see 2.28. This is quite a substantial standardized difference. Let's look at the point estimates. The trial says a 12% increase in DVTs in patients with pulmonary embolism, comparing rivaroxaban versus endoxaban in low molecular weight heparins. While we find this protective effect 0.67, what's going on? We're going to talk about this in a minute. The last batch, osteoporosis medication, Horizon Pivotal, Vero, chronic kidney disease, stabbed by CKD, heart failure, asthma, and COPD, kind of the respiratory package down here. And it's getting quite checkered, and you see here already that we have difficulty emulating these trials in design and in their measurement. And you see also quite some differences now, -3.4, -5. Where you have an RCT finding of 0.85, a 15% reduction in COPD exacerbations, versus we do see a 13% increase. What's going on here? So let's dive in. And I can tell you that in this process, I learned way more about randomized controlled trials than I ever wanted to know. But it's quite an interesting world. I happen to not do RCTs. I love RCTs, but clearly there's also quite some interesting things going on in the world of RCTs. Now, these are the 2 trials where we set out to predict, which means we deposit the finding before the trial results are released. As in diabetes, the Carolina trial, linagliptin versus sulfonylurea, where we were quite close. Prostate cancer pronounced with regard to a MACE endpoint, major cardiovascular side effect, and we were quite happy with those results, and we felt good about this from the beginning, that we were able to emulate these trials quite well. Okay. Now we went through the 32 trials, and let's talk a little bit more about the emulation challenges. Now, use some examples, and so that we can come to some lessons learned. There's a whole bunch of emulation challenges that we go through. Let's first look at the inclusion-exclusion criteria. Now, this is a typical list of inclusion-exclusion criteria of a trial. This is Paradigm HF. This is for Entresto in heart failure patients. And there's a whole bunch of criteria that we can emulate quite well. This is in black here. Sufficiently emulated that is in yellow. Difficult to emulate is in red. There are certain things like presence of any disease with a life expectancy of less than 5 years. All right, how do you quantify that in an RCT? Certainly, I have no idea how to quantify that in secondary data. There's a lot of subjectivity in some of these criteria, right? In other measurements, we simply don't have. HFrEF, HFpEF differentiation is difficult with the data sources that we have available. Now, but for 2 characteristics, we could emulate the inclusion-exclusion exactly, which is age and sex. And look what we see. Actually, totally as expected, that RCTs have way fewer women included, than in clinical practice databases. And participants in RCTs are younger on average than in clinical practice. Although we have the same age range defined from the lower to the upper, we still have a different distribution of age, which is more to the older age in clinical practice than in the randomized controlled trials, right? Now, keep in mind, you could re-rate according to this. We did not re-rate because, remember, we set out very narrowly, had the investigators pre-specified a real-world evidence study, what would the results have been, right? This is not about re-weighting and making them even similar to RCTs. This is post-hoc, and we are doing this later on. But for the primary analysis, we just emulated the inclusion-exclusion criteria, and this is what we found, okay? Not a big surprise, although I was a bit surprised sometimes about how few women were included in randomized controlled trials. So the comparator emulation and placebo controls, we classified in 3 categories: good, moderate, and poor. It's kind of semi-qualitative, obviously. And here's some examples where these trials, the BETIS trials, LEADER, DECLARE, EMPA-REG, and CANVAS, were all placebo-controlled large cardiovascular outcome trials of these second-line antidiabetic medications. Now, since we don't have placebo, we picked DPP-4 inhibitors as a comparator. These are also second-line antidiabetics, and they are not associated with 3-point MACE improvements with the cardiovascular endpoints. That any trial that was done didn't show any improvement in cardiovascular endpoints. So in a sense, they are working like a placebo because based on the best evidence that we have, they have no effect on the outcome, okay? Now you see that actually it works quite well, 0.87, 0.82, 83, well, 6.9, not so well, but qualitatively moving in the same direction, at least showing the protective effect of GLP-1 receptor agonists and SGLT2 inhibitors. 0.86, 0.83, and so on, right? So this seems to work reasonably well, okay? Now here's another example where we emulated placebo. Now for these trials, the DPP-4 trials, which was our comparison group, remember, in the previous trials, because we thought with regard to MACE, they shouldn't have any effect. Carmelina, TICOS, Sabatini, Carolina, and all of them showed no effect against placebo in the trial. Now, we use sulfonylureas as a comparison. These are penny-cheap second-line treatments that are around for decades, and we do see this slightly protective effect. We think that this is residual confounding because of the economic selection that is ongoing. The copay is substantially higher in these newer medications, and you might have risk selection, that economic risk selection, which we know is on average associated with BMI, physical activity, and smoking, all risk factors for cardiovascular events. So we do think that this is residual confounding. Outcome emulation, what we do see in the RCT, particularly when we have low specificity of the outcome assessment in the real-world evidence study, that the event rates are usually event rates are higher in the RCT, but if we have lower specificity, then, which means we are falsely assuming that some of the events are outcomes, although they are not really, right? So the randomized trials have an event rate, for example, of 21 and 26 here, while we have 46 and 44. We have double the event rate. Clearly, our events are diluted with non-events, right? Because we have a definition that is not specific enough. In-hospital start of medication, we're working with claims data here, which means we don't know what medications were given in the hospital during hospitalization. Triton, Timi, Plato, ESA-REACT, these are these antiplatelet trials. They start in the hospital. You have an ACS, acute coronary syndrome. They get started right away. And what we see when you look at one of the publications here in Medium, Viviot, you look at the Kaplan-Meier and zoom in on what's happening at the beginning. The effect is at the very beginning. This is still in the hospital setting or peri-hospital setting, the space that we don't observe. Well, after that, there's no effect anymore. There's no effect anymore, a very mild effect. It's exactly what we observe. We observe exactly this tail here of the Kaplan-Meier, but we miss the effect size at the very beginning. Hence, we have different findings. We don't emulate this very well, and we have then also quite different findings. Delayed effect with a long follow-up period. So this is Horizon Pivotal. This is one of the osteoporosis trials where we compare a once yearly bisphosphonate infusion, zoledronic acid. The trial went on for 3 years, and you see this effect size is 40% reduction in the risk of hip fracture. And in real evidence study, this is the Kaplan-Meier real evidence study, we see this 0.75 is only a 25% reduction. Now, qualitatively, they're moving in the same direction. The clinical interpretation is the same, yet it's still a substantial difference in effect size. Now 36 months, the hazard ratio for 36 months is unobservable in our data. And the reason for that is, turns out after the first injection here, a year later, nobody comes back. In clinical practice, I think out of these 9,000 and 9,000, 18,000 people, I think 3 or 4, a handful of people got a second injection, a second infusion somewhere around here, which is why we had to truncate follow-up here, because nobody exposed anymore after half a year here. But it turns out, if you look just at the first 18 months in the trial, which is now the same 18 months that we have emulated in the randomized, in the real-world evidence study, it's actually spot on. 0.75, 0.75. So we clearly have a valid study. We have a valid study when we compare equal to equal, the first 18 months versus the first 18 months. But we fail to emulate the overall trial because in clinical practice, the adherence patterns are such that we will never get to the adherence that's enforced in most randomized controlled trials. Okay. Now, if you, this is the Horizon pivotal point here, this would basically move straight to the diagonal based on what I just showed you. We didn't do that because we stuck with our predefined prospective protocol for all of these studies. But I'm just saying, right, this is part of the learning, right? Run-in windows, many trials have run-in windows, and based on the experience in run-in periods, some patients get ejected from a trial, right? This is particularly frequently happening in the respiratory space. And this is an example here. That is the, what is the trial? Where is the trial? Impact, sorry, the Impact trial. It's actually 2 trials packed in one, where it's all about triple therapy for COPD, ICS, LAMA, and LABA. The baseline therapy is a 2-week run-in period. But the interesting thing here is, and then the comparison group is just a dual therapy, one ICS and LABA and LAMA-LABA combination. Now, turns out that 40% of subjects were already on triple therapy at baseline. So almost half, 40%, were on triple therapy, like this box here. They had the same as in the dark blue box over here. Now, how do you get into this arm? You take something away that they were happy on, apparently, because they landed in the trial being on triple therapy, and you take something away. Well, if you take something away, well, the other side looks probably good. And Sammy Sweet has written for the last decade on this scheme that we see in these respiratory trials. And that, of course, leads exactly to these beneficial effects in the trials because you take something away, something that worked for the patients. And of course, you don't observe that in clinical practice. You simply don't observe that in clinical practice. We see flat null findings for both of those arms because if you don't have this weird selection, you actually have an outfinding. Triple is just as good as dual therapy in clinical practice. And I'm quite convinced that our finding is valid. We're studying something quite different from the randomized controlled trial, which is a takeaway trial rather than an adding trial. That makes us study 2 different things, right? Which doesn't mean that the real evidence estimate is wrong, quite the contrary. I think this is actually really a valid finding for clinical practice. And you see, of course, in the trial, an Impact trial, that all the effect is just at the beginning, when the first 30 days, when you have this acute effect of taking something away. And in the second 30 day, the whole show is over and it's flat, okay? Now, what you see from that is, if you put this together, and this was quite a dense run through of these issues, what differentiates what is done in RCTs versus clinical practice, that we do quite well in emulating these pragmatic designs. The more pragmatic the trials are, the more they reflect use practice of like from clinical practice, the more they mimic dosing patterns. If they don't have run-in periods and things like that, the better we can emulate these trials. The more and more constricted the design of the trial gets, the harder and harder it gets to emulate them with data from clinical practice for all the obvious reasons that we just went through. So as we are looking at this again, you see the trials that we emulated quite well, the trials that we have difficulty emulating, and their correlation coefficients. And then you have these binary markers for regulatory agreement. That is a binary marker based on p-value smaller than 0.05. So this is the FDA's regulatory mark, and we may agree or disagree with that arbitrary binary cut point, but for them so far that's a holy number. And if we would apply that, actually in the well-emulated trials, we would have in 100% regulatory agreement, we'll come to the same regulatory decision, right? So that's quite encouraging. Now there's a problem still. If you look at this here, for example, what about this Einstein PE? Right, that's an ISO. Wouldn't it be much nicer if this was gone and everything would be really nicely aligned here? So let's look into that a little bit. And this is, and Einstein PE is part of the Einstein program, Einstein DVT PE, same hospital centers involved, same protocol, same outcome assessment, open label, both of them. It's just these are subjects were recruited who had DVT. These are subjects recruited who had PE. The outcome is the same. It's recurrence of DVT. Okay. Now, why is that? And here are the point estimates. You see, we do see this protective effect in the Einstein DVT. We can emulate this quite nicely, 0.68, 0.75. The upper 95 crosses the one, where you see the ridiculousness of a binary p-value 0.05 statistic as an agreement. So the asterisks mean that we still hit the non-inferiority, which this was designed for. But the problem is this here. Einstein PE showed this 12% increase in risk of DVT, while we continue to see this protective effect, very similar to the Einstein DVT, right? We were quite consistent. It turns out, actually, there was a meta-analysis. Meta-analysis also showed no heterogeneity between a PE population or a DVT population. The effect size was always very similar. And this shows the natural variability that you see in trials. A single trial is not necessarily the truth, even if it's completely validly designed and conducted and all that. And the most recent example, of course, hit headlines was Aduhelm, right? The Alzheimer medication, where they had also two trials that were exactly likewise designed, and still one was statistically significant at 0.05 on whatever metric, while the other one was a flat null finding, was as null as it gets, right? So that is an issue when you do these emulation exercises. You emulate against one RCT, right? And you assume that is your gold standard, but we know, of course, that a single RCT is still not the truth. But there's no way around that, because we want to emulate a specific study as closely as possible. Well, let me conclude here. The real-world evidence studies come to the same conclusions as RCT when we emulate the design and measurement of the trial well, right? And I hope I've convinced you about that. And this is the blue dots in these diagrams. This is just convince us, yes, we can come to the same conclusion if we were interested in exactly the same question. Now, of course, real-world evidence is there to complement RCTs, not to make away from RCTs. So actually, we want to study questions that are actually different from the RCT question, which is the red dots. The red dots, I would argue that most of them are, if not all of them, highly valid studies, and they are most likely true findings to a question that is slightly different from the question that the RCT was answering. We are unhappy with this, with the agreement statistics. They were predefined with FDA. We spent months in agreeing on this, agreeing on the agreement statistics, but there are many more different statistics, and different stakeholders want to have different metrics for success or failure. In evaluating real-world evidence study in principle, right, independent now of this replication activity, it is enormously helpful to always think about an RCT. How would an RCT look like in order to answer the question? Because then you see very, very quickly discrepancies in the design and measurement between the RCT that you contemplate, you might never do this RCT, versus the implementation that you do in your secondary data, and where you think you need to compromise here and there, right? But overall, it's a very upbeat message that we think we produce here, and there are many more follow-on investigations. One of them being the use of electronic health records in order to get into fields that we cannot touch with claims data. Think about lab test results, think about functional status improvement, think about cognitive status improvement, skin, difference in the skin lesions over time, and all sorts of other markers that we don't observe in claims data. But there's hope that we can do better with electronic health record in combining those two data sources. So let me pause here and take questions, and then I can certainly expand further.
Terrific. Thank you, Sebastian. You can stop sharing your screen, and we'll go into picking your brain a bit further. So let me start with a couple questions for you. The first one is a bit in the weeds, and then I'll get a little broader. But I noticed in your many of your tables that the effect sizes you were trying to replicate were had confidence intervals that crossed 1. And given that there's an issue of significance there for those, and therefore less surety that those are in fact real effects, how enamored are you of those effects, those particular effects, in terms of trying to replicate them? Is that even something you feel is worthwhile pursuing?
Yeah, no, that's a great observation, Ken. And indeed, it turns out that particularly when the investigators set out with a non-inferiority hypothesis, and they actually, in my mind, they found a superiority, but the 95 was just still spilling over the 1. And in many of our real-world evidence emulation studies, because these databases are so large, right, and that's what you're getting at, right, we had way more power, right? So our 95s are much narrower often, right? Yes. And if you then take 0.05, we are spec significant, while the trial is non-significant, which is why this regulatory agreement statistic is really quite dissatisfying. It makes much more sense to really focus on the point estimates and separately look at the uncertainty that is around it, looking at the 95s, rather than having this one p-value metric with this binary cut point.
Great answer. So, and then I wanted you to just opine for a moment on a related discipline, which is this sort of replication of trials or in vitro trials with synthetic data. There's a lot of momentum in that space, and looking at the issues that you are teasing apart with great care and concern around real data, real world data, what is your assessment of the utility of using synthetic data for, for example, adding a synthetic arm to a trial or some of those objectives?
Well, that's something of great importance. Lots of discussions around this. It starts with define synthetic. So what I call this external control arm, right? And there are a variety of data sources that you can use as an external control arm to a single arm trial, right? And often enough, it is electronic health records, and sometimes it's registry data that people use. The difficulty there is, on top of all the issues that are described here, the difficulty you have is that the single arm trial, by definition, is primary data collection, because you have an experimental drug, you're in the field, you have primary data collection. And the reason why people do these external control arms is because they want to save money, or they argue they cannot find enough subjects to randomize in a trial. But there you have secondary data. You have secondary data, so you compare primary data versus secondary data, whether it is historical data or concurrent data, and that is leading to this differential information content in the two sources, right? As you know, primary data are, you can define what you measure, when to measure, and how you measure, but in secondary data, somebody else is doing all that for you, and you've got to live with that, right? And often enough, that this is quite different. And Brad Hamill from Duke had just published something comparing the primary data from a trial against electronic health records and against claims data, and that's quite discouraging, particularly with the electronic health records, actually. With claims data, actually, he's closer to the primary data. So that is additional level of complication. I'm not saying this doesn't work at all. It works, but the first look that you have and that the FDA has is at the data source. What is the secondary data source that you use for external control on, before you even go into anything that we discussed here, the analytics and things like that.
So I'm going to go to some of the questions that come from the participants, and folks can put additional questions in using the Q&A box down at the bottom of the screen while we get started. So first question's from Felix Dietlein, one of the CHIP faculty. Thank you so much for this great talk, Dr. Schneeweiss. I learned so much. I had two questions. From a cancer-focused research perspective, I was wondering whether emulated trials might be generalizable towards multi-omics data. Many investigators in my field publish their source code or protocol and update or expand data after publishing their study. Could rerunning their code on updated data identify biases at an even earlier stage, for example, before a biomarker or drug enters a clinical trial? There's the second part of the question, which is, to what extent are differences between original data and emulated trials expected by random chance? I suppose some difference would be expected by chance, and the baseline might vary between fields and diseases. Is there a good way to quantify the contribution of stochasticity to this effect?
Wow. Okay. So let me start with the second one. That's easier for me to understand than the first one. This second one, yes, absolutely. We would just by chance expect differences, which is quantified in this metric. It's called standardized difference, which is the difference between the point estimates and the standard errors considered in the 2 point estimates. But that does not quantify the inherent variability within RCTs that are going beyond the 95. And you see this in the Einstein PE versus Einstein DVT example that I showed you, where these two trial findings were so far apart. So this is only a partial quantification. It's an interesting point that you raised, whether that quantity differs by therapeutic area and things like that. That's I cannot comment on that. It's we can hypothesize on this, but with the 30 trials, I don't think we can quantify that. I'm not sure that I fully understand number one. So if Felix is on the,
let me see. I'll see if let's start with your excellent answer to number two.
Yeah.
we'll move on. Felix may clarify in the Q&A. This is from Jay Min. Dr. Schneeweiss, thank you for the engaging presentation. We started in the beginning with how one programmer may interpret a protocol differently than another. Do you find that a person may emulate an RCT protocol differently than another person emulating the same study?
Oh, excellent question. Yes, that's absolutely right. So there is another activity ongoing. It's also funded by the FDA, the CERSI programs, where two groups were tasked to emulate the same trial, and they didn't talk to each other, and they came up with slightly different parameters that they picked. And that resulted in minor differences. These were only emulating two trials in those two examples, but that is a good point, which is why we need to be rigorously transparent in what we do, so that your audience can identify exactly what choices you made, right? And but nobody is really protected from that. We are all part of that, and we need to be transparent. That's the only protection that we have. And by the way, that programmers interpret the same protocol in two different ways. You could also turn this around and say, hey, just write better protocols. Clearly, your protocol was ambiguous, right? So there's no blame that you want to distribute here. This is a shared learning. We all need to do better.
Right. Here's one from Lokendra Thakkar. Is it possible to construct a weighted stratified index for emulation challenges, which could then be used for RCT versus real-world evidence evaluation?
Yeah, and so when we published the first 10 emulations, and the main paper is not yet out, still stuck in peer review, we got a letter, it was circulation, we got a letter, and the letter writer was saying, well, the most important finding here is really the difficulty in emulating trials. Was zooming in straight on the emulation part, which goes to your point here. Can we better quantify emulation? And we are currently doing a meta-regression trying to find out which, we have 6 parameters of emulation success broken down in different aspects of the study design and measurement issues. Which aspect of the emulation closeness or distance is driving these results most, right? So that is quite informative. Keeping in mind with this whole conversation about emulation success, this is relevant if you really want to mimic an actual RCT, right? Because, but if you do a real-world evidence study without emulating an RCT, you emulate a hypothetical RCT, your study question might be perfectly fine defined. So the emulation thing is really just a thing if you have an actual trial. Otherwise, emulation differences become a secondary issue.
Right. And Felix has responded that largely you actually answered that question on your last slide. And he had put that in before. Yes. So, Dr. Schneeweiss, this has been really informative. You presented so much data in such a clear way in such a short period of time. I think it's both very encouraging that there's a real scientific discipline emerging here that you're at the cutting edge of, that will clearly have an impact on regulatory processes, and also that some of the questions about it or conceptions that those of us here on the Zoom might have had a priori may have shifted a bit, and I think one area where you really emphasize is it really may be that this emulation and duplication, as it were, in RCT-DUPLICATE is a very nuanced, should be a very nuanced endeavor where you have to be very careful about which questions you ask and which questions, not just to duplicate the trial, but to figure out and not just to complement the trial as it is, but to actually ask even different questions than the trial was going to ask originally, and that these things really need to fit together in ways that perhaps are more nuanced than perhaps than was conceived of
Absolutely.
5 years ago. Go ahead.
Yeah. So this rigorous causal inference thinking really helps and makes studies better together with transparency. What we also learned, of course, when we sifted through the RCTs, we realized this tremendous shortcoming of the data source that we're using. So we've seen not a single arthritis trial, for example, where functional status is the key outcome, which we simply don't observe, right? And so the next phase of this is to complement the claims with electronic health records. And that, I think, is also where the FDA is going with the Sentinel Innovation Center, where we build larger and larger data source, where you have both linked together. And you marry the clinical granularity from the EHR data with the care continuity of the longitudinal data of the claims data, right? So there's great hope that we can expand the types of questions that we can answer with these secondary data in a valid way.
Great. Thank you so much for spending the hour with us today, and for the work that you're doing. I'm going to just briefly share my screen to let folks know about our upcoming talks in the Intelligent Health series and also in the Landmark Ideas series. Coming up just after Thanksgiving on December 5th is the inventor and futurist Ray Kurzweil. After the new year, we have Richard Miner, inventor of the Android operating system, Alan Brandt, who is a extremely inspiring professor of history and medicine at Harvard University, Ron Balliser, who drove much of the real world evidence work at Clalit around the many topics, but especially the Pfizer vaccines, with world-shaking studies last year. Julie Gerberding, who is at the National Institutes of Health and formerly directed the CDC. Karen Copenhaver, who is a internet and e-commerce lawyer who will talk about the role of open source licensing in a very nuanced way. Robert Langer, who's Institute professor at MIT and the most cited engineer in history. Ian Lipkin, a extremely well-known virus hunter who always has good stories to tell. Tom Mayer, who the medical director at the NFL Players Association, who made football much safer for players these last 7 years. Molly Ann Brody, who is a pollster at the Kaiser Family Foundation, who will talk about the role of polls in health policy, and Christina Farr, formerly health and technology reporter for CNBC. You may have seen a lot of her work in the past, and now she's a health tech investor. And her work there is informed by her prior role as a reporter. So thank you all for spending the time, and come reach out to us at the Computational Health Informatics Program, www.chip.org, and we hope to see you at the next seminar. And thank you again, Sebastian.
Thank you, Ken.